Hello students, I am Mrs. Ghodake-Patil working at Swami Ramananda Bharati Vidya Mandir and Junior College Tasgaon. Today we study the cell organelles from cell structure and organization. These different cell organelles that we are going to study today, that is the Golgi bodies, mitochondria, microtubules, that is cilia, flagella, nucleus, ribosomes, and chloroplast. All these cell organelles are important structures or these important cell organelles. So we start from the Golgi complex. This Golgi complex located near the endoplasmic reticulum in the cell. It acts as a packaging transport unit of cell and consists of number of stacks of membranous sacs called as the cisternae. So see in the diagram, these are the cisternae. That means this tubular part is known as the cisternae. It is of different size and shape. There are number of, that means they are few to several, and it depends on the function of the cell or function of that organelle. Thickness and molecular composition of the membranes at one end of the Golgi sacs differ from those of the other end, because these are the different ends. These contents or these sacs shows the specific orientation in the cell. Each system or each cell has a forming phase, or it is known as the cis phase. This one is the cis phase and this one is the trans phase. Without this, it shows the transport vesicles. These are the secretory vesicles and newly formed vesicles, and these vesicles that pinch off from transitional endoplasmic reticulum and merge with the cis phase of Golgi cisternae, and its contents into the lumen. अशा पद्धतीनं हे स्ट्रक्चरल ॲक्च्युली स्ट्रक्चर आहे गॉल्जी बॉडीचं. ह्याच्याशिवाय आपल्याला ह्याचे फंक्शन स्टडी करायचे आहेत. The functions are different and important in the cell, that are the secretion, modification of secretions of endoplasmic reticulum and production of own secretions. त्याच्यामध्ये सिस्टर्नी कंटेन्स स्पेसिफिक एन्झाईम्स फॉर स्पेसिफिक फंक्शन, म्हणजे विशिष्ट अशा एन्झाईमचं विशिष्ट फंक्शन आहे. कुठलंही एन्झाईम कुठलंही फंक्शन परफॉर्म करत नाही, तर देअर इज द स्पेसिफिसिटी अँड रिफायनिंग ऑफ प्रॉडक्ट्स टेक्स प्लेस इन ए सिक्वेन्शियल मॅनर्स. आणि त्याचं एक्झाम्पल आहे sugar components is added or removed from the glycolipids and glycoproteins, and that are brought from endoplasmic reticulum and thus forming a variety of products. त्याचे जे ओन प्रॉडक्ट्स आहेत किंवा ओन सिक्रेशन्स आहेत इन मेनी प्लांट सेल्स प्रोड्युसेस cellular polysaccharides like pectin. Pectin is one of the most important polysaccharide like the cellulose. Manufactured or modified all products of Golgi complex leave cisternae from transphase as transport vesicle. That is important. अशा पद्धतीनं Golgi bodies with their functions आपण स्टडी केलेली आहे. नेक्स्ट सेल ऑर्गनली, that is the lysosomes. It is also important cell organelle. Lysosome, see in the structure, single wall membrane. It is the single wall membrane and enzyme complex system. There are list of enzymes, number of or several enzymes secreted by the lysosomes. We study lysosomes are dismantling and restructuring unit of cell, and it contains number of hydrolytic enzymes. These enzymes digest macromolecules, and the enzymes also shows optimal activity in acidic pH. Lysosomes arises from the Golgi associated with the endoplasmic reticulum. And besides this, there are list of lysosomal enzymes includes hydrolases. That enzymes is a hydrolytic enzymes, and the list of hydrolases enzymes is amylases, proteases, and lipases. Besides this, lysosome is known as the polymorphic nature, or it shows the polymorphism phenomenon because it secretes the primary vesicles or primary lysosomes, secondary lysosomes, or these are known as the hybrid lysosomes, residual body, and autophagic vesicles. किंवा आता आपण वन बाय वन बघतोय. प्रायमरी लायसोझोम्स आर द मेम्ब्रेन बाउंड व्हेसिकल्स in which enzymes are in inactive state, and secondary lysosomes are also known as the hybrid lysosomes, which are formed by fusion of lysosome with endocytic vesicles containing digested material and represented at heterophasic vesicles like this. Thus, see in the diagram, it is the another diagram with functions performed by the lysosome, that is the polymorphic in nature or polymorphism phenomenon. This is the plasma membrane, food entered through the plasma membrane, then it forms phagocytosis, food entered, this food get digested. Digestion of food material takes place and remnant remains as in. Thus, see the residual body. It is a vesicle containing undigested remains left over in the heterophasic vesicles and after releasing the products of digestion in the cytosol. म्हणजे सायटोप्लाझम मध्ये अशा पद्धतीने रिमेन्स पाठवले जातात. Lysosomes brings about digestion of cells own organic material like damaged cell organelles. स्वतःची डॅमेज झालेल्या सेल ऑर्गनलीज जे आहेत त्याचं डायजेशन स्वतः करतं आणि म्हणून अशा ॲटोफॅजी किंवा ॲटोफॅजिक वेसिकल्स फॉर्म केले जातात आणि म्हणून हे फंक्शन आहे डायजेशन करण्याचं ऑर्गॅनिक मटेरियल रिमेंट्स झालेलं. त्याच्यामध्ये डॅमेज्ड सेल ऑर्गनलीज ज्या आहेत त्याचं डायजेशन होतं म्हणून त्याला सुसाईड बॅग्स म्हटलेलं आहेत. म्हणजे लायसोझोम्सचे दोन कॅरेक्टरिस्टिक आपण बघतोय. एक आहे that is the polymorphic phenomenon, and besides this lysosomes are commonly known as the suicide bags. Then see the functions of lysosomes. These brings the intracellular as well as extracellular digestion. For example, intracellular digestion is brought about by the autophagic vesicles or the secondary lysosomes by the process phagocytosis. And extracellular digestion is a release of lysosomal enzymes outside the cell, and well-known example is acrosome. It is secreted by the sperm and it contains an enzyme hyaluronidase, and that enzyme useful during the fertilization process with the egg, sperm and egg union, that is dissolve the protective layer of the ovum. One more function, that is the lysosomes play important role in the metamorphosis. That means the lysosomal enzymes helps in the tissues of redundant organs. They also helps in the destruction of malignant cells, and that example is T lymphocytes. There are a number of different types of cells in the blood or blood corpuscles, and that is the T lymphocytes. तर अशा पद्धतीने लायसोझोम एक इम्पॉर्टंट सेल ऑर्गनली आहे. Then we study one more important cell organelle, that is the mitochondria. See the structure of mitochondria. It is somewhat oval in shape. The shape may be variable. Then this organelle shows outer membrane, inner membrane, intermembrane space, then matrix filled in that membrane, DNA, then cristae, finger-like projection of the inner membrane, and it shows the ribosomes. Thus, number of contents are present in this, but these mitochondria are useful in or play a important role in the aerobic respiration. These are absent in prokaryotic cells and RBCs. These are the double membrane bound cell organelles. Outer membrane is permeable to different metabolites due to presence of proteins present in it, that is the porins and Parsons particles. Inner membrane is selectively permeable to few substances. Only the selective particles are permeable through the membrane. Both membranes are separated by the space, that is the outer chamber, and inner membrane encloses a cavity in a chamber containing matrix. Matrix contains coils of circular DNA, RNA and 70S ribosomes, lipids and various enzymes of Krebs cycle and other pathways. तर अशा पद्धतीनं स्ट्रक्चरल और मेम्ब्रेन बाउंड सेल ऑर्गनली जी आहे that is known as the mitochondria. Commonly these are known as the powerhouse of the cell because it synthesizes the ATP for the different parts or for the different activities of the cell. And one more part is important, which are present on the cristae. See, these are the cristae and these are having the particles. That particles are known as the oxygens or F1 particles. These play important role in the synthesis of ATP. Membrane of mitochondria bears number of particles that are known as the oxysomes. These oxysomes shows head region and foot region that contains stalk and base. Each particle or the head faces towards the matrix and base faces embedded in the inner membrane. Head acts as an enzyme ATP synthetase, and foot acts as the proton channel. Besides this, the function or oxysomes involved in function in proton pumping and ATP synthesis. That is the source of ATP synthesis, and also it shows the or regulate the metabolic activities of the cells, and it promotes the growth of new cell and cell multiplication. अशा पद्धतीनं mitochondria are the important cell organelle. Then next cell organelle, that is the plastid. These are also double-walled organelles containing DNA, RNA and 70S ribosomes. Plastids are again classified according to the pigments present in it, and these are leucoplast, chromoplast and chloroplast. Leucoplasts do not contain any pigment, and they are useful in the storage of nutrients. Chromoplasts contain pigments like carotenes and xanthophylls, and they give red, yellow and orange color to flowers and fruits. And the chloroplast is found in mesophyll of leaf, and these are oval, spherical, discoid or ribbon-like. Thus, cells may contain single large chloroplast in Chlamydomonas algae, or can be 22 to 40 chloroplast per cell in mesophyll cell. Actually, we study the structure of chloroplast. See the diagrammatic representation. It is green in color because it contains green pigment called chlorophyll, and it shows, or these are useful in the photosynthesis. So see the structure of the chloroplast. In the chloroplast double membrane, it shows the external membrane, also internal membrane, then space between intermembrane space, then plastoglobule, ribosomes, thylakoids, the coin-like structure that is known as, or these are connect with the granum. These are the thylakoid plate-like structure, the lumen of the thylakoids, then lamellae, then grana and stroma, that is the liquid part in between them. Thus, actually, the structure that we are going to study, the chlorophyll, that is the green pigment with enzyme that help in the production of sugar by the process known as photosynthesis. That means chloroplast plays important role in the production of food material by the process of photosynthesis. Inner membrane of double-walled chloroplast is comparatively less permeable, and inside the cavity of inner membrane there is other set of membrane called membranous sacs, and that are called as the thylakoids. That are the number of sacs known as the thylakoid. Thylakoids are arranged in the form of stacks. This is the stacks, and these are known as the grana, singular granum. And these grana are connected to each other by the stroma lamellae. Space outside with the thylakoid is filled with the stroma, and the space inside the thylakoids contain different enzymes essential for the photosynthesis. That means simply the chloroplast are plays important role in the process of photosynthesis. Stroma of chloroplast contains DNA and ribosomes. These are the ribosomes, and DNA, chloroplast DNA is present in this. Then next cell organelle, that is the nucleus. Actually, it is the important cell organelle. It is controlling center of the cell. The nucleus of eukaryotic cell becomes distinct in non-dividing cells during interphase. Cell division मधली एक phase आहे that is known as the interphase, and such interphasic nucleus is made up of nuclear envelope, nucleoplasm, then nuclear membrane न्यूक्लिओलस अँड क्रोमॅटिन नेटवर्क. तर अशा पद्धतीनं आपल्याला त्याचं स्ट्रक्चर बघायचं आहे, पण त्याचे कंटेंट्स बघा. फर्स्ट, न्यूक्लियर एन्व्हलप. इट इज द डबल मेम्ब्रेन or double walled limiting membrane of the nucleus, and two membranes are separated from each other by perinuclear space. Outer membrane is connected with the endoplasmic reticulum and inner membrane is lined by nuclear lamina. That is the network of protein fibers that helps in maintaining shape of the nucleus. Then nucleoplasm or karyolymph. It contains various substances. See in the diagram. This is the ultrastructure of nucleus, and it shows all the contents: outer membrane, then inner membrane, then there are the openings that are known as the nuclear pores. Then in between these outer and inner membrane there is perinuclear space. Then heterochromatin, then chromatin threads. These are the chromatin threads. These are the chromocenters in between the threads. Then karyosomes and karyolymphs. Besides this, there is the nucleolus. Thus, the nucleolus made up of rRNA and ribosomal proteins, and it is the best known as the site of ribosome biogenesis. These are one or more nucleoli present in the cell. Then nucleoplasm. न्यूक्लियस मधलं जे सायटोप्लाझम आहे त्याला न्यूक्लिओप्लाझम असं म्हणतात. They appear as a dense spherical bodies present near the chromatin network. न्यूक्लिओलस हे क्रोमॅटिन नेटवर्क जवळ दिसतं आपल्याला. They produce rRNA, ribosomal RNA and ribosomal proteins which are then transported to cytoplasm and assembled together to form ribosomes. तर अशा पद्धतीनं रायबोझोम फॉर्मेशनमध्ये रोल महत्त्वाचा आहे. देन क्रोमॅटिन नेटवर्क. All these thread-like network that is known as the chromatin network, that is the contains genetic information in the form of chromosomes which contains DNA molecules and associated with proteins, and the chromosomes appears in the thread-like network that is known as the chromatin material, and that chromatin material contains DNA, histone and non-histone proteins and also RNA. अशा पद्धतीनं क्रोमॅटिन नेटवर्क जेनेटिक इन्फॉर्मेशन कंटेन करते, त्याच्यामध्ये त्याचा रोल फार महत्त्वाचा आहे. आणि काही रिजन्समध्ये क्रोमॅटिन DNA इज मोर अँड जेनेटिकली ॲक्टिव्ह कॉल्ड ॲज युक्रोमॅटिन, अँड ॲट सम regions that contains more proteins and less DNA, and these are genetically inert called heterochromatin. अशा पद्धतीनं युक्रोमॅटिन आणि हेटेरोक्रोमॅटिनमध्येही जेनेटिक मटेरियल डिवाइड केलेलं आहे. The cells prepare to divide. The chromosomes coil and get condensed. Coiling form होतं, and metaphase स्टेजमध्ये they becomes distinct and clearly identify. हाही बदल सेल डिव्हिजनमध्ये होत राहतो. काही फंक्शन्स दिलेले आहेत, म्हणजे number of functions performed by the nucleus, but important are they contains entire genetic information, hence play important role in the heredity and variation. It contains, or it is a site for synthesis of DNA, RNA and ribosomes. It play an important role in the protein synthesis, and also as it contains the chromosome number, it constant for a species, thus it is important in the phylogenetic study. And thus nucleus is the master cell organelle. Important cell organelle आहे आणि त्याला master cell organelle असं सुद्धा म्हणू शकतो आपण. Next cell organelle, that is the ribosomes. Ribosomes are protein factories of the cells. They use genetic information to synthesize proteins. इथंही रायबोझोमचा रोल प्रोटीन सिंथेसिसमध्ये फार महत्त्वाचा आहे. Ribosomes are made up of ribosomal RNA and proteins. They do not have any membrane or covering around it, and it is made up of two subunits. One is large and one is small. See in the diagram. These are the ribosomes, which contains one smaller unit, subunit, and one larger subunit, and entire ribosomes made like this. Then actually prokaryotic cell मध्ये 70S type ribosome असता. It is made up of 50S and 30S subunits, and eukaryotic cell मध्ये 40S आणि 60S अशा पद्धतीचं smaller आणि larger subunits असतात, and it forms the total entire of 80S, S for Svedberg unit. Then actually these are ribosomes in the cytoplasm are either found attached to outer surface of rough endoplasmic reticulum and nuclear membrane, or sometimes they may be freely suspended in the cytoplasm. These are two types: bound ribosome, which produce proteins that are transported outside the cell after processing in endoplasmic reticulum and Golgi bodies, and free ribosomes come together and form chain called polyribosome for protein synthesis. Both type of ribosomes can interchange position and function, but one more important function of ribosome, that is play active role in the protein synthesis. Then next cell organelle, that is the cytoskeleton, and in cytoskeleton we study the structure of cilium and flagellum. Actually cytoskeleton is supportive structure built from microtubules, intermediate filaments, and microfilaments. Microtubules are made up of protein, tubulin, and microfilaments are made up of protein, actin, and intermediate filaments are composed of fibrous proteins. So see the structure of flagellum. They help for, or cilium, help for locomotion in unicellular organisms. त्याच्यामध्येच त्याचा रोल महत्त्वाचा आहे. इट कन्सिस्ट ऑफ बेसल बॉडी, बेसल प्लेट, and shaft. Basal body is placed in outer part of cytoplasm and derived from the centriole, and it has 9 peripheral triplets of fibrils. These are total 9 triplets. And shaft is exposed part of cilia and flagella, which consist sheath and axoneme. Sheath is covering membrane of cilium or flagellum, and core called axoneme possesses 11 fibrils or microtubules running. See here, these are the 9 triplets and these are the 2. Thus 9 plus 2, 11 fibrils. And central tubules are connected to each other by a bridge enclosed by the central sheath. Thus the cilium or flagellum actually plays an important role with the peripheral doublets by a radial spokes. Wheelचे स्पोक्स असतात त्याप्रमाणे स्ट्रक्चर दिसतं, अँड कनेक्टेड इच अदर थ्रू लिंकर्स. लिंकिंग आहेत तिथं प्रत्येकामध्ये, आणि त्या लिंकिंगमुळं त्याचं स्पोकसारखं स्ट्रक्चर फॉर्म झालेलं आहे अँड डबलेट ब्रिज फॉर्म केली जाते. त्याच्यानंतर नेक्स्ट सेल ऑर्गनली आहे सेंट्रिओल और सेंट्रोझोम. सी दिस स्ट्रक्चर इज लाईक द कार्ट व्हील. गाडीच्या चाकाप्रमाणे स्ट्रक्चर दिसतं इथंही आणि इथंही. ॲक्च्युली हे सिलेंडर्स हॉरिझॉन्टल किंवा पॅरलल फॉर्म केलेले परपेंडिक्युलर टू इच अदर. सी द स्ट्रक्चर अँड देन हाऊ इट फॉर्म्स. ॲक्च्युली इट प्ले सिग्निफिकंट रोल इन द स्पिंडल ॲपरॅटस ड्युरिंग सेल डिव्हिजन. स्पिंडल फायबर फॉर्मेशनमध्ये त्याचा रोल महत्त्वाचा आहे. अँड इट इज जनरली लोकेटेड नियर द न्यूक्लियस इन द ॲनिमल सेल. कंटेन्स सिलेंड्रिकल स्ट्रक्चर्स कॉल्ड सेंट्रिओल. हे सिलेंड्रिकल स्ट्रक्चर्स आहेत. These cylinders are perpendicular to each other and surrounded amorphous substances called pericentriolar material. These connect with each other and forms 9 sets of triplet microtubules, which are made up of tubulin, like in the cilia and flagella. These triplets are spaced triplets connected to each other by non-tubulin proteins. There is a connection with the non-tubulin proteins. And the proximal end of centriole, there is a set of tubules called as the hub. And in that hub, actually they shows the spokes of the radial spokes. And these are due to these proximal ends of centriole looks like a cartophyll-like structure. This is the cartophyll-like structure. Thus, centriole forms basal bodies of cilia and flagella. That's all about the all cell organelles which are important for the cell. Thank you.